So it's a small group. I think I know, not quite half the people here. But just by way of introduction, my name's John Bachman. I'm a research fellow at the Lab of Systems Pharmacology at Harvard Medical School. I'm here with my colleague Ben Gary, and he and I have been working on all of the projects that I'm going to describe today together. And so one of the benefits of giving the last talk of the day is I get to kind of put my slides together at the last minute and make it sound like I've solved all the problems that have come up during the meeting. And so, so I actually wanted to, rather than bury the lead here, I wanted to kind of give you. I summarized some of the points that I think that we address in some of the tools that we've developed that I think are relevant, in particular to this audience. So just at a high level, I would say that the stuff that we're developing are a set of tools that are useful for building very large but also detailed causal models. At large scale is redundant, but I guess I mean sort of literature scale or genome scale. And this involves a cluster of related technologies. So on what you might think of as the curation side, we've developed tools that can use NLP that allow biologists to curate statements or mechanisms in English. We can use NLP also to extract statements from the scientific literature, and I'll talk about how we do that. We can also translate information between, from and to different existing pathway formats. And that includes biopacked content, and we can read from BEL. We can also assemble back into BEL by virtue of our integration with PyBEL, as well as many other formats. One of our big focuses is the assembly and quality control of large corpora. These are things that arise when you start to try to bring mechanistic information together from many databases as well as from many different natural language processing systems, so I'll talk a bit about that. We have tools that can use BEL and other types of mechanistic content to build different types of causal or executable models. We've built a framework for literature-scale text mining and knowledge assembly, including storing the information and processing it in the cloud. And rather than become a kind of provider of a platform, sort of the next pathway commons, we've included the software for these tools in our open source package, so that if you have access to these reading systems, you can set up the same kind of infrastructure in the cloud yourself. And finally, something I'm also going to talk about is a framework for building domain models that self-update based on reading the literature as they appear. So with that sort of some key bullet points out of the way, I thought I would offer some background as to how we got involved with the BEL community and to some extent sort of why we're here. So I did my PhD in Peter Sorger's lab at Harvard Medical School, and the Sorger lab has been working for over a decade on systems biology models coupled to experimental data, in particular dynamical models, kinetic models of signaling systems. And so here are some of the key papers. These are from 2008, 2009, and these are sort of what figures in these papers might look like. So you see, you have a model. This is from a model that involved about 500 ordinary differential equations. These are time traces from the model with experimental data from different cell lines, and the idea was to use these kinetic models to explain this kind of experimental data. And the models that were being built in the lab at that time looked like this. This is a set of ordinary differential equations in MATLAB. And as we were working with these models, we understood that there had to be a better way to represent mechanistic information that usually came from papers or domain knowledge in some other form, and turn into something that was actually transparent, right? So this is completely inscrutable to anyone other than the person who wrote it, and even to that person if they come back to it a few months later. And so one of the things I worked on in my PhD was to develop a software package called PySB, which aimed to use elements or concepts from computer programming and software to make better models. So rather than representing things as sets of ordinary differential equations, you can write macros and functions that are tailored to your domain, and then you can write high-level models that draw on and recombine those elements that serve as a kind of domain-specific vocabulary for your model. And so it was with that in mind, with this idea of drawing on software concepts, that we started thinking also about testing models, unit testing models, and collecting qualitative observations about biology and testing to see if your model can recapitulate that behavior. And we got wind of BEL, actually through Walter Fontana, who's another professor at HMS. And we started looking at the role of BEL, or the use of BEL, is because it captures qualitative observations. It gives us a way of saying, okay, here's a list of a whole bunch of things that should happen upon treatment with EGF. Now, which of those things can our model recapitulate? And we were working on that, and it was about that time that the DARPA Big Mechanism program appeared. And this was from the website describing the program. It says, the Big Mechanism program aims to develop technology to read research abstracts and papers, to extract pieces of causal mechanisms, assemble these pieces into more complete causal models, and reason over these models to produce explanations. The domain of the program is cancer biology with an emphasis on signaling pathways. So this is very relevant to what we're doing and also relevant to what people in here are doing. And we were fortunate enough to be funded under this program, and that sort of set our work off onto the direction that I'm going to focus on for the rest of this talk. So initially, though, we focused, again, on BEL, but now not only as a source for model constraints, but also for information about mechanisms, right? So if you think of a corpus of BEL statements, you have the direct statements, which essentially represent the direct physical molecular mechanisms, and then you have the indirect mechanisms, which represent these kinds of high-level observations or constraints. And our very first thing that we did was write a translator that took direct BEL statements and basically translated them into macros in PySB, right, the software that I described before. And in fact, the software was called BelPy. And in fact, if you go to GitHub sorgerlab/BelPy, it will redirect you to our current software project. And later on, we actually got wind of PyBel, and we thought, oh my God, someone's done the exact same thing, but then it turned out to be something different. But so that the reason it's not called BelPy anymore is because early on we realized that we didn't want to just draw from BEL. There's lots of different types of sources we wanted to draw on, including pathway databases, which involves BEL, as well as other formats. The scientific literature, as you can see, that was one of the things we had to focus on in the context of Big Mechanism, and also potentially domain expertise, curated knowledge in one form or another. And then all that information had to come together into some kind of intermediate knowledge representation before it could then go out again into one or more different model formalisms. And the reason we think of modeling as different from knowledge representation is because it's been our experience, as biological modelers, that models tend to be very use case specific. We call them fit to purpose models often. And whether you want a detailed dynamical or kinetic model with perhaps a small number of variables so you can do parameter estimation versus a large directed network that at the genome scale really depends on the kind of data you have and the kind of question you're asking. So we wanted to separate out the process of gathering information about mechanisms and the process of assembling models with this intermediate knowledge representation. And so that's the conceptual structure, and this is what the software architecture of Indra looks like. Indra is the name of the software that we've been working with, or that we've been building. It stands for the Integrated Network and Dynamical Reasoning Assembler. This actually is it's an outdated diagram. There's actually many more nodes on both sides of this diagram here. These represent different knowledge sources. It includes BEL, which is the oldest in Indra, but also different natural language processing systems like Reach and TRIPS, also for BioPact. All of these sources of information are normalized into Indra statements, which I'll talk about. And then from there, there's different assemblers that can render that information in different ways. And in the middle, there's this crucial layer where you operate on statements themselves to identify relationships between statements and quality control the kind of corpus you have. So I mentioned Indra statements as a kind of knowledge representation. I wanted to talk about how they differ from BEL, and this is something that we realized we needed to do early on. If you think of the statement like mech phosphorylates ERK, for example, how does BEL approach a statement like that in terms of how it's represented? In BEL, everything in biology can be represented with a causal relationship of an increase or a decrease, to a first approximation, right? There's other types of relationships as well, but mainly it's an increase-decrease type relationship. Whereas in biopacks, it takes a different view, which is that everything in biology can be represented as a reaction, right? You have inputs, and then a reaction happens, and then you have outputs. The trouble is, if you think about it, let's say you read a paper and you see that an author says that MEK phosphorylates ERK. In both cases, the high-level biological semantics is actually lost. So it may be true that in the paper there was an observation, a kinase assay that showed that a particular kinase and its activity increased the abundance of the phosphorylated substrate, which is how you effectively represent it in BEL. But the assertion, if you only have the text to go on, that assertion, you have to essentially reinterpret it back into an experimental observation to represent it as an increase in BEL. Does that make sense? Right? So you would turn that into kinase activity of MEK increases directly phospho-ERK, basically, is how that would be represented in BEL. Whereas in biopacks, you'd have MEK plus ERK and on the left as reactants, and then MEK plus phosphorylated ERK on the right-hand side. The thing is, if you take that literally as a reaction mechanism, it's a very simplistic one. In fact, depending on what you're modeling, what happened to the enzyme substrate complex, for example? How do you model the effect of a drug, that kind of thing. And so what we realized we had to do is capture these high-level semantics that really are a good impedance match to the kinds of things you get from the literature, these kinds of assertions, and implement a broader taxonomy of these kinds of relationships, and then subsequently handle how those things are converted into a model at assembly time. So in Indra, for example, that's just phosphorylation, MEK, ERK, and that's the closest as we can get to the original source material that we have. So these are some the types. This is basically a summary in UML of the types of statements and entities that we can represent in Indra. It's heavily focused on signaling and post-translational modifications, but it also can be used to represent some metabolic reactions as well as transcriptional regulation. One key thing is that we can incorporate a lot of information about agents or biological entities. They can have different types of state. They can have modification or mutation conditions. They can be bound to other proteins, and they can have different types of activities. And then we have a model for storing evidence about these relationships. So one trade-off here that should be mentioned up front is that we don't we didn't go into this trying to develop a universal knowledge representation for biology. We really focused on modeling applications, and so there are areas of biology that we don't cover or high level. We don't have a lot of emphasis on high level disease processes, although we can model them in principle. So it'll be interesting as we talk later about the BEL language and going forward with enriching the BEL language. It'll be useful for us to sort of think about how we can extend our representation as well. So that's what that's how we capture the information and normalize it. So just briefly, I wanted to mention the kinds of sources that we can draw on. So I mentioned pathway databases. We can draw from anything in biopacks. We can draw from BEL. But typically, we just work with the BEL large corpus. That was the only source of BEL content that we were aware of for a long time. We recently discovered causal bionet, actually, and have been working on drawing on that now that we can access the JSON formatted files through PyBEL. We also have found Senior to be a very useful resource. This is a curated signaling pathway resource of about 11,000 reactions, and BioGrid is really good for physical interactions. And I also mentioned different natural language processing systems. So the key here is that we are actually not NLP developers, although we've learned a tremendous amount about the field through collaborating with a bunch of different groups over the last 3 or 4 years. And our approach has really been to try to make use of as many NLP systems as we can get our hands on, because we find that the approaches tend to be quite different. The types of errors that these systems make are quite different, and by assembling information extracted by these different systems together, we can get much higher quality output and also more coverage of the available of information in the literature. So the systems that we currently interface with are the TRIP system, developed by IHMC in Florida, the REACH system, developed by the University of Arizona, Sparser, which is developed by a company here based in Lexington, Massachusetts, TEAS is an event extraction system that is, I think, relatively well known in the academic side of BioNLP. And they've, I think, scored well in the BioCreative BEL challenges and so forth. We recently developed an interface to MedScan output through collaboration with Elsevier. GeneWay's output, which is a system developed by University of Chicago, and a new system we've just been developing recently, the ability to handle results from this system developed by USC. So we're constantly trying to add new systems so that we can assemble information from these different sources. And the next one actually is we're going to actually be meeting with Sumit to talk about how to think about incorporating belief into this system going forward. So that's how we collect information. So I want to talk about assembly. So something that often I think, and a metaphor that I think often is helpful to people in biology is to mention that knowledge assembly is a lot like genome assembly in the sense that you have some sort of underlying truth, some kind of template information, but the information that we can get from the literature is sort of fragmented and noisy and involves a lot of overlaps and duplicates. And so you might imagine that, like, if the underlying truth is that phosphorylated MEK phosphorylates ERK at this particular site, we get a bunch of different, we have, MEK phosphorylates ERK, okay, that's pretty good. We've also got methylethyl ketone phosphorylates ERK because it's misgrounded it. We've got ERK phosphorylates MEK because the reading system transposed the subject and object. We've got a site that's inconsistent with the reference position on the protein, and so on and so forth. And the question is, how do we pull together all this information, address these errors, and try to get as close to this unified representation as possible? So the first thing is that we had to address some of the systematic errors that came in from both reading and databases. And the one that I wanted to just focus on is this issue of grounding, which means finding identifiers for named entities encountered in text. So in particular, we found that families and complexes were often not grounded by these reading systems or were incorrectly grounded. So for example, we found that NF kappa B, this is the ranked list of the most frequently encountered ungrounded entities, and in this program at DARPA, which is actually focused on RAS biology, RAS itself was not grounded, right? So that's a problem. And mTORC1 is a complex, actin is a family, NF kappa B is a family of complexes. Right, so these are common among the ungrounded entities. And the ungrounded entities were distributed largely towards the frequently encountered ones. So we realized that we could do some curation, and we developed a resource that we've called FamPlex for families and complexes and their lexicalizations. It's available on GitHub. It's under the CC0 license, so really it's free to anyone to use and redistribute and remix, and hopefully the community will find it helpful. We have a preprint up on bioRxiv. This paper should be coming out hopefully soon. We just resubmitted the second round of revisions a few days ago. And just as an overview of the structure of the resource, FamPlex consists of a set of resources, basically CSV files that link named entities like AKT with a set of identifiers which are defined in this FamPlex namespace that define families and complexes. It defines equivalences to those, and then it defines their members effectively. So this should sound familiar to anyone who's worked with the Solventa families and complexes. I'll sort of get to that in a moment. But one of the things we showed in the paper was that integrating these identifiers along with these synonyms that we curated empirically from the literature that we read improved grounding of families and complexes dramatically, from something like 20% to something more like 80%. One of the key things here, and this is something I was discussing with Natalie over lunch, is that we found that in many cases these entities were neither strictly families nor complexes, but actually you might call them famplexes, or a complex of families or a family of complexes. So if you take AMPK, that's a holoenzyme, which is a heterotrimer consisting of 3 different subunit families: the AMPK alpha subunit, the beta subunit, and the gamma subunit, each of which has multiple genes. And we have just 2 relation types, ISA and part of. And we found frequently, actually, that in the literature, these event extraction systems were pulling out information at all 3 of these levels. So, you had information about AMPK, but you also had, dexamethasone upregulated AMPK alpha phosphorylated at 3 and 172. And for this assembly task, it's actually important to identify these hierarchical relationships. So because we have this ability to recognize relationships between genes and families and complexes and their subunits, we can actually arrange statements into a hierarchy, not just based on family to member relationships, but also based on their statement specificity. So, for example, this is a very generalized statement about the phosphorylation of MEK by RAF. This is more specific because it involves individual genes. This has information about the site, and up at the top you have the most specific versions of this statement, which indicates both the residue and the position of the phosphorylation. So what we do is we collect information from many sources, we organize it into this kind of hierarchy graph involving a set of hierarchies that we've built for entities, modifications, locations, and activities. Now, one thing that also came up today was this issue of how to score or apply some kind of measure of confidence in an edge in one of these networks. So we think about this problem of believability largely in terms of the likelihood that something is a reader error, because the biggest source of error that we deal with is error introduced by the reading systems, because the precision is anywhere between 50 and 80%. So we have a lot of errors. But if you think about it, if a reading system has a particular probability of getting an extraction correct, then the more times you see that sentences describing that mechanism, and the more times you extract that same mechanism, the more confidence you have that all of those extractions are not jointly incorrect. And so what we do is we calculate the joint probability of an incorrect statement given repeated extractions from different sentences. We can also do this by combining results from different readers, which may have different error profiles and different precision profiles. And then in addition, we propagate these error estimates through that statement hierarchy graph that I mentioned. So for example, if MEK phosphorylates ERK2 at T185 is true, then it is necessarily true that MEK phosphorylates ERK2. So if this is true, it by itself may not meet a threshold of belief 0.95, but by virtue of its having a probability of 0.89, it makes this statement more likely to be true. So we do this belief propagation process in a component that we call the belief engine. And at the end of the process, we end up with scores assigned to each of the statements in this graph, and then we can slice off the layer according to our precision recall trade-off for our application. So I just wanted to now mention some applications of this kind of technology. And the first one is, I think, for people involved in curation might be interesting, which is essentially model building and through natural language itself. And this is actually our first paper that we wrote about the Indra system, was published in Molecular Systems Biology, and it was basically focused strictly on this use case, which was building computational models from natural language. We showed some examples of how you could build kinetic models of the activation of P53 in response to different types of DNA damage using a set of simplified natural language sentences. And there was a very nice news and views written about our paper by Jeremy Purvis, actually, who titled it. He titled his news and views, "Put Your Model Where Your Mouth Is," which I think is a kind of a great concept in terms of if we think about the word models that biologists routinely traffic in, and how there is a barrier to getting those word models into something that's executable and testable. And here's a more complex example describing ERK reactivation after vemurafenib treatment. You can see that we can say things like SOS1 bound NRAS binds GTP, GTP bound NRAS that is not bound to SOS1 binds BRAF. So actually the NLP is good enough to pick up some of these complicated subclauses and conditional relationships. I don't know if I have time for this, but I thought I might. I had a short demo to sort of show how this can actually be used. What's that? Let's take the time. Take the time. So if someone give me a molecular mechanism in words.
A whole one. I'll let somebody else.
Anyone? Well, I have some that I know work.
Go for it.
All right. So this is just yeah. Well, so this is the script, and this actually it might be helpful to see. So the Indra, the software architecture, is pretty nicely modular. So I'm using PyBel here. I'm using the TRIPS NLP system from Indra.sources, and I'm using the PyBel assembler from Indra.assemblers. And then in terms of the actual action here, it's 1, 2, 3 lines. And Charlie, I had an error here. I had to do this to get this to work. I had to populate document.name. But anyway, it's really 3 lines. We process text with trips. We get a trips processor, which contains Indra statements. And then we assemble those Indra statements with a PyBel assembler. We make a model, which is now a PyBel bell graph. And if I say something like sauce1 bound to EGFR activates KRAS, for example. It'll take maybe 10 seconds here. This is going out to the TRIPS web service. So you can see, I've gotten a BEL document out in BEL script, which is now deprecated, as I understand, but you could.
No. Wasn't dealing
So you see here's the header information, and here's the sort of the information you're looking for. We have a complex protein HGNC SOS1 and protein HGNC EGFR directly increases the activity of HGNC KRAS, for example. And it's also added in the information that this complex has component SOS1 and so on and so forth. One other one that and this is what the Indra statement looks like. It's an activation statement with SOS1 bound to EGFR and activates KRAS. There's another one that and no one wants to offer one. I'll offer another one. Here you have, let's say, and I'll play with the names. Let's say I'll say MEK1 phosphorylated at serine 218 and serine 222 phosphorylates. Let's say ERK, which is the family this time, at T185. So instead of saying threonine 185, I'll say T185. So here we have P-HGNC MAP2K1. So you see it normalized MEK1 to MAP2K1. It's got the dual protein modifications at 218 and 222, directly increases. It's been grounded to the FamPlex namespace, which we have equivalences to the Solventa family, so it actually could have been mapped to that if we had done that at 318 and 185. So I don't know how useful this is because BEL is already pretty unlike, let's say BioPax, which you can effectively not write in any kind of human readable way. BEL is reasonably tractable for human curators, but I think it is an interesting use case to contemplate in the context of visual editors, incorporating a kind of natural language option. And we've
actually created kind of a BEL glitch where we take BEL and turn it back into natural language-ish.
Yeah, actually, in fact, we can we'll do that as well. We have a thing called the English assembler, where we can take Indra statements and assemble them back into English, which is really useful for rendering it on figures and curation interfaces and things like that.
John,
yeah.
I also stepped one at the top level API for PyBel, so you can just go right from PyBel and use the English assembler back to.
Oh, when you use the Indra English assembler?
Yeah.
Oh, cool. So I'll go back to slides here. But actually, one of the things that came up earlier is this idea is like, why can't journals have authors create machine-readable nanopubs out of their publications? And we've had some interesting discussions with Chris Sander and Gary Bader. Chris Sander is at the Farber now, and Gary Bader is at the University of Toronto. We've been working apparently for many years on a system called Factoid, where they essentially want to create a tool that will do exactly that. They want publishers to get licensed to software, and then when someone publishes a paper, they get a link that's already been processed with some event extraction systems that sort of say, we think this is what's in your paper. Would you like to add anything? If they want to add anything, they can express it in natural language and things like that. So we've been talking with them about using this kind of a tool to incorporate natural language input into a system like that. So other applications, so there's 3 other applications I wanted to mention. So this one actually is probably the most familiar to this audience, which is to use literature mining and aggregated pathway databases to build large causal networks of causal mechanisms, to use those causal mechanisms as a set of prior knowledge for data analysis. And this is another application that we've been pursuing. One of the key things, though, is that, as I mentioned before, we can assemble different types of models. So depending on our application, the type of data we have, it could be sort of very coarse-grained directed protein interaction graph, a Boolean network. an influence map of an executable model. I won't go into too much detail as to what that is, but the point here is that different models live on a continuum, I think, of in terms of their mechanistic detail and causal context. I think of that largely in terms of how much detail is in the nodes. So actually, BEL and PyBEL would actually be between these two things, because BEL explicitly represents like node state, node active modifications, mutations, activities, not just individual genes and their activities as they would be in a directed graph or a logical network. And so in an application, there was one application we worked on recently. We used this representation, which is where we actually build an executable model that could be simulated dynamically, but we actually analyze it statically. And the way that we do that is we create this influence map, which is a representation that can be derived from the kappa rule-based modeling system, where the nodes are actually events. Think of them as mechanisms, and then the edges represent activation or inhibition relationships between mechanisms. So the binding of DUSP to MAPK1 actually activates the dephosphorylation of MAPK1 by DUSP, which in turn inhibits the abundance of the phosphorylated protein. So what we did is, in an evaluation exercise in Big Mechanism, is we took a data set published by Chris Sander, which involved 140 proteins and phosphoproteins measured by reverse phase protein array. And we looked for causal paths linking particular drugs and drug targets to those measured proteins. Now, some of them, so we want to actually come up with hypothetical causal paths for how that could have happened. And I won't go into too much detail, but this was our procedure for building the model. We read about 100,000 papers with 3 different NLP systems. We pulled information from Pathway Common and a large corpus. We processed it. We did this kind of assembly and quality control. We ended up with a model with 220 proteins and 1,450 rules. These were the 22 effects in the data set with the largest fold change. And as you can see, some of them are sort of trivial, right? So that if you inhibit MEK, phosphorylated ERK goes down, right? So green means that we were able to find a causal path, which is good, right? Because MEK and ERK are actually immediate neighbors to each other in the graph. And we actually found causal paths for all of these that were in green. So 20 out of 22, we struck out on CDK4, the effects of CDK4 on these proteins. But for these other ones, we were able to find candidate causal paths, right? In this context, because the data is not sufficient to discriminate like what is the correct causal path. But the idea was that these causal paths had sufficient detail and sort of the causality was actually transitive in them, such that A activates B implies that B activates C, right? And so when you went back to curate these mechanisms, you could see that it actually logically made sense. So for example, here's an example of one that's sort of non-trivial. How does SARC inhibition increase this particular phosphoprotein? Here's just a simple graphical representation of the pathway, but under the hood, it's all Indra statements, which have a lot more information. So it says SARC phosphorylated on tyrosine 418, phosphorylates PAC2 on S20. PAC2 phosphorylated on S20, so that's the conditional relationship between the two, that's sort of the antecedent and the consequent. Phosphorylates RAF1 on serine 338, so on and so on and so forth. And you go through here, there's actually this relationship between p53 negatively regulating PLK1. And the nice thing is that all of these are annotated with evidence from the literature. So if you look in the evidence for this, this is the URI from Pathway Commons, but the REACH reading system also pulled out these, it was about 10 sentences from about 7 different papers, indicating clearly that p53 represses PLK1, p53 dependent repression of PLK1, p53 decreases PLK1. So that was something that I hadn't heard about, but actually was seemingly quite legitimate. In the discussion we had earlier about how do we enrich things that curators curate, mechanisms that they curate with, how do we find the evidence for those statements? We found often that you can take a model and then even if you don't use these natural language processing systems to make your model bigger, you can use it to attach evidence to all of those mechanisms that are in your network because of this ability to find duplicate and hierarchical relationships between statements. looking at our sort of overall performance on this particular data set, you can actually see that when we looked at smaller folded changes, we had 95 out of 135, and actually we did pretty well on all of these drugs, but very poorly on these 3 drugs, something we still haven't really haven't managed to figure out exactly why the network that we had or the algorithms that we had weren't able to find causal paths between these 3 explanations for these 3 drugs. But that's something that we're looking into. And sort of a minor note here is that if you want to make additions to your model, you can actually make additions to your model in natural language, as I showed. So just now quickly, because I'm going over time, because the systems that we built are almost entirely automated or highly automatable, one of the things that we were able to do is create models that update themselves by monitoring PubMed for literature as it appears, reading it with a set of reading systems, and then adding to that model and finding what the difference is, what the mechanistic delta is, because it knows what information it's already read and can sort of assemble that into the stuff that's already there. So as an example, we built sort of a software agent called that we call the RAS machine, which is a self-updating network model of RAS. It's hosted on Index. If you go to the Twitter handle for the RAS machine, there's a link to that network. And the idea is that every day it has a set of keywords, and every day it looks for literature related to those keywords, which involves genes and a few other biological terms, diseases, that sort of thing. Finds new mechanisms, assembles them into the network, updates the network, and then it gives you an update, to tell you that it's running and that it read a certain number of full text papers and abstracts and learned some number of new mechanisms. This is something that we have generalized to other biology areas outside of RAS. We put up, not in addition to the RAS machine, the diabetes machine and the heart machine. Actually, Charlie has used this technology to create to get running a Alzheimer's machine. And we're looking to really broaden this collaboration with Charlie's group and also Dexter Pratt's group at UCSD to try to bring a kind of more rich and robust front end to these networks so that curators and domain experts can see the updates as they appear. And lastly, just something that is perhaps not as relevant to this audience, but something that we've been doing as part of another DARPA program called Communicating with Computers, is integrating these kinds of knowledge-based systems into dialogue agents. So a few years ago, Triadica published a paper called Theory of the Cell, which is kind of a think piece imagining what if we had intelligent assistants or intelligent agents that could help scientists and doctors sort of answer questions like these. In estradiol-treated SKBR3 cells, which nuclear protein complexes have the greatest change in phosphorylations? And the long-term vision would be that systems like this could be embedded within molecular tumor boards to kind of, when you're sort of off the textbook route of evidence-based medicine and you have doctors who are trying to reason about an individual patient's mutational profile and therapies and trials that are available, a system like this could be part of that dialogue and help formalize some of the reasoning that goes on and actually draw on this corpus of knowledge that it has by processing the literature. These are some of the kinds of questions that it could be used to address. So we've been, for now, we've been focusing on questions that are not unlike the example that I showed with the Corkett et al. Data set, where we say, we use the system to try to find explanations between a perturbation and an observed effect. So for example, why could this drug, PLX4720, result in an increased amount of FOS? This is actually real dialogue from this system, which is called Bob. You can build a model with Bob. You can say things like PLX4720 into its BRAF, which goes out to this natural language processing system, as I showed briefly kind of in the BEL demo. And this is just a snippet of this dialogue. I'm not going to show the rest of it, but we have some more examples we can talk about later. But the idea is that we've been testing this in the lab with some of the computational biologists to see how do they do with this system versus just sort of googling around and poking around databases. And it's anecdotally currently appears to be getting to be where it's actually quite effective. So we're working on that. We'll be working on that project for another couple of years. So in summary, we built a system called Indra that builds many types of mixed models from many sources. This knowledge assembly process involves correcting, merging, and filtering large numbers of mechanistic fragments. we can do this kind of natural language modeling, which we typically do to build small models or do curation. Large models can be extracted from the literature and used to explain effects in large data sets. The automation of this process allows pathway models to self-update. New analytical methods, I think, will be needed to make use of causal models that are both large and detailed. That's actually what I was very interested in. We were very interested in Eric's talk today because we're looking for ways in which these types of very rich priors can be incorporated into statistical and data analysis pipelines. The website's at Indra.bio. In terms of future plans and sort of the things that we're working on right now, we're always looking to make these tools bigger and better, more reading systems, more sources of content, more databases, more taxonomies. We're actually working to expand the system to domains beyond molecular biology. We're actually funded under a grant called World Modelers to make this effectively open domain. The advantage for biomedicine is that that will actually give us the capability to move to diseases and biological processes and higher level concepts that are not strictly, don't rely as much on the taxonomies that are available for molecular biology. We're working to use these networks in statistical inference, to do causal explanation of large data sets, to expose these biomachine or RAS machine capabilities as a web service to different types of consumers, including index, but possibly others. To integrate Indra as a kind of back-end knowledge engine into analysis and curation interfaces, which could include index, BEL commons, pathway commons, factoid, that system that I mentioned earlier, and others. So if you were interested, we'd love to collaborate with you. A lot of people worked on this project. I mentioned Ben. This has been an equal collaboration. We've also worked with a number of other groups, and all the funding for this was provided by DARPA. Thanks.
So, as an anecdotal point, the notion of molecular tumor boards was actually, we did that for 3 different tumor boards using Foundation Medicine's diagnostics. We presented mechanisms and pathways. The trick is, you show all, 100 potential tumor-involved genes, it's overwhelming.
Yeah.
So the trick is to find a way to condense it for a particular class of patients. So it turns out you can take of the genes you showed and full, let's say, hundreds of % of them, you can squeeze them to 20 modules, meaning you have the signaling components, you have a few of the pathways that cascade, and then you have various ways of working with the nucleus. If you turn those into little visual icons, and of course, when the patient has a mutation there that's been well identified, the module's red. And when you click on it, you see the actual mutation. It allows you to compare different patients much more accurately. And then, of course, you're right, you embed where the therapies currently work. It became a very major talking point whenever they showed this, because in the room there's usually 1 or 2 specialists in molecular biology, and the rest sort of know bits and pieces. Having it there live and interacting with the cases changes the dynamic right away. And so that was a proposed product to be done. It didn't go that way, but it's still, I think it needs to. In fact, UC Davis made it clear that they didn't get that module in some form. They were going to cancel the contract with Foundation.
Very interesting.
So it's very, very timely, I would agree, to do that.
Okay. Very cool.
What's funny is we did something like that back in 2009 with Hopkins. Mm-hmm. And the challenge that we saw at that point, because we would identify which mechanisms were activated or inhibited in the patients, is that most of the discussion of the tumor board was, do we want to do broad chemotherapy or chemoembolization? The use of targeted therapies on tumor boards is very limited.
It's changing. It takes a long time, but the number one thing is not the science, it's the reimbursement. Unfortunately, that's changing now. It's been a problem.
So about the text mining tools.
Yeah.
So when we visit the biocreative workshops and also the biocuration workshops, the biotherapy workshops, so the message there is no way around assisted curation. So because the scores of text mining patterns are not good enough for the biologists yet.
Yeah.
Also, if you aggregate several models, machine learning models, the score just rises 2 to 3%.
Yeah.
So how are you coping with this problem if you are just using automated curation?
Yeah. So.
This
Yeah.
experience is also in our department,
Yeah.
Also of PMIs.
Yeah. I mean, I wouldn't necessarily claim. So one of the ways in which we aim to address this is, again, we don't build a hybrid model. We essentially we rely on numbers and sort of brute force to some extent to separate signal from noise. So that's one part, right? So reading more literature makes a big difference. Most of the things I've seen in the bio-NLP world, people process the PMC-OA corpus and that's it, and they're done, right? But we've worked on getting, we've got all the content from PMC-OA, we've got the PMC authors manuscripts, we've got everything in Medline, and we've also are working to integrate Springer and AAAS content as well. Because again, the more chances you have to see that mechanism in a form that reader will understand, the higher your chances that you'll get.
But if you get full text from all these sources, I think you get another problem because text is just not text plain documents.
Yes,
so
that's another.
tables, figures.
That is, yes. For some of these, all the ones I mentioned so far, we have structured XML content. We are only just now dipping our toe in the water with PDF, extracted PDFs, and you're right, it's not pretty at all. It's pretty awful. So we're hoping that by the time we get there, eLife will have solved this PDF extraction problem with their deep learning-based, image analysis algorithm. But another approach that we're looking at for that is just to do the PDF extraction and then find ways to triage documents that turn out to be garbage and filter them out, just so we don't choke the reading system, because that also happens. But the other key piece is also using multiple readers. So you use multiple readers and you can use them to corroborate each other. And we
also
actually
databases, by the way.
What's that?
Databases. So in some of these
Yes, that's true
applications, we
too.
pull in the BEL large corpus and pool of pathway comments with relatively high confidence scores, and those are mechanistically aligned against the machine readers, and often make a big difference in how believable that makes, in a technical sense, the readings.
I have also a hypothesis, which you can tell me whether it's true or not, but in some of the things I've observed in the bio-NLP community, I feel like there are certain scoring criteria that matter to that community that don't matter to many sort of biologists in the context of data analysis. Like, I was looking at I was in an evaluation we did for FamPlex where we were looking at accuracy of grounding of families and complexes. We looked at the BioCreative 6 named entity annotation data set, and you were scored wrong if you saw, ERK in a paper and you grounded it to human ERK when it was actually mouse ERK, you were scored as wrong. It was no different than like saying it was a completely other protein. And that meant that people spent a whole lot of time trying to, do this full discourse analysis to figure out, okay, are we talking about mouse or human? But 99% higher, 99.9% of the time, I think biologists don't care.
Yeah.
And I think that there's a lot of that going on.
Yeah, it's only the
But
case
that's just a
when-
theory.
When the species detection is important.
Yeah.
I think when BAL challenge, we actually ignore
Yeah.
which kind of species you are detecting.
Yeah.
So we use actually the orthogonal
Yeah.
genes and proteins. Yeah.
But like, this is another thing that came up is like, for example, if something describes a transcriptional upregulation and you list it as an activation, meaning a regulation of activity instead of a regulation of amount, should that be scored as wrong, or should you consider it as sort of a superset of the other? I think that there are things like that we're actually right now in the middle of doing an actual quantitative evaluation of the role of multi-reader assembly to see how much it matters to read with more readers. So I'll have numbers in like 3 weeks.
That's currently I'm doing also to evaluate not only the relation extraction tools, but also all these relation extraction tools contain sometimes the same name entity recognition because there are not a lot of tools outside available. And they are making the same errors every time. So if you are aggregating all the results, you're actually aggregating all the errors too.
That's also true, which is why because of that, we had to integrate this systematic error component in our probability model, which is to say, certain readers, let's say there's let's say the rule-based information extraction systems. If there's a bug in the rule, like it's interpreting statistical associations, like alcohol is associated with liver disease, and it's taking that as a physical interaction, for example, it's going to get that systematically. And so you have to look at what is the probability of a statement still being incorrect, even if you had, 1,000 instances of that extraction. And there is some probability of that can be parameterized as well. And that's sort of the way we have a random error component and a systematic error component. So that's how we deal with it. Yeah.
For these kinds of statements, have you tried to divide your corpus up into different types of documents? For example, review articles may be more rigorous on terminology than scientific reports, or even looking at books.
We haven't yet. We've been focusing on articles so far. As we, again, try to get more content, we are interested in looking at like gene riffs and Wikipedia articles and other sources of content. We're just right. We've been building. We spent a lot of time. Ended up building. When things scaled to not being able to run and be stored on a single machine and in a Python pickle file, we had to spend a lot of time building infrastructure for storing this information in a database and running it in the cloud. And we're just now finishing that up to where we can now start to draw on other corpora and look at the differences between the type of source and what essentially the error profile is for different types of content. It's something we haven't modeled yet.
I was going to mention one other interesting causal connection, or maybe simply correlation, but Doug Laufenberger was on the board of GenStrike for a number of years, early days.
Oh, cool.
Yeah.
Thank you. Why don't we take a bio break and
A BEL.bio break?